The overall goal of this method is to synthesize a magnetic nanoparticle inside a protein cage and then to functionalize the protein such that it enables rapid attachment of the magnetic nanoparticle to cells.
This method can be used to generate magnetic nanoparticles that enable rapid and efficient magnetic labeling of cells, which is important for applications such as MRI or magnetic cell separation. The main advantage of this technique is that cell magnetization can be achieved using low nanoparticle concentrations and short incubation times, which prevents potential adverse effects due to nanoparticle exposure.
To begin, deoxygenate 500 milliliters of deionized water by placing a tube connected to a nitrogen gas cylinder into the water and sealing the vessel with cling film. Then, bubble nitrogen gas through for approximately 60 minutes. Heat a water bath connected to the double-jacketed reaction vessel to 65 degrees Celsius. Then, add 75 milliliters of 50 millimolar Ikebasque buffer pH 8.6 into the reaction vessel. Seal the vessel and deoxygenate by bubbling nitrogen gas through the buffer solution for approximately 20 minutes. At the same time, stir the buffer solution using a magnetic stirrer. After deoxygenating the Ikebasque buffer solution, remove the nitrogen tube from the buffer, keeping it suspended over the solution to maintain a nitrogen atmosphere. Add apoferritin to achieve a final concentration of 3 mg per milliliter.